Hello viewers, today we are going to understand the structural morphology of lymph node, specifically its microscopic features. As you all may be knowing, lymph nodes are small bean-shaped glands that are located along lymphatic system. The lymphatic system is a system of vessels similar to arteries and veins through which lymph fluid travels. Lymph nodes are classified as secondary lymphoid organs, with the primary lymphoid organs being the thymus, tonsils, spleen and bone marrow. Now you are seeing the afferent vessel, which takes lymph towards lymph node, and efferent vessel, which takes lymph away from the lymph node. Now let's see the internal structure of the lymph node. In this video, the lymph node is bisected at the level of efferent and afferent vessel along with the lymph node parenchyma. You can see this is the afferent vessel, as I have already mentioned earlier, carrying lymphatic fluid towards lymph node. The structure you are seeing is the capsule made up of dense connective tissue. The inward extension of capsule, which divide lymph node into different compartments, is known as trabeculae, and the space between capsule and trabeculae is called subcapsular space. The lymph node is broadly categorized into two portions. The outer portion of the lymph node is called as the cortex, and the inner portion is called the medulla. The dark area that you are seeing now is the cortex, which is abundant in B cell, and surrounding these cortical areas is the paracortical area where T cells are abundant. Now this is the medullary sinus, where medullary lymphatic fluid are present. Here B lymphocytes aggregate to form primary follicle. When there is antigenic stimulation of B lymphocyte, primary follicle convert into secondary follicles. As we know, primary follicles have lymphoid follicles without a germinal center. On the contrary, secondary follicles have lymphoid follicles with a germinal center. Primary follicles mostly contain B cells, whereas secondary follicles mostly contains lots of T cells, B cells, plasma cells and antibody. Now you are seeing comparatively lightly stained area in the center of a follicle called the germinal center. As germinal center has less chromatin content, so it is lightly stained. This is all about lymph node histology. Hope you understand well. Thank you viewers. Please do subscribe and share my video if you like them.